Hello everyone, welcome back to AS and A-Level Biology with Dr. Demi. I am Dr. Demi, and in this video I am going to show you the solutions to Paper 5 2014. Now for many of you that might seem like an odd paper to choose because it seems like it was so long ago, but what I have found is that this question paper in particular tests your knowledge of how to apply, it tests your ability rather to apply your knowledge to things that you're not familiar with. So many of my students have gone through this paper and for them it was not a pleasant experience, but I hope that by doing it here you would start to see or get comfortable with the unfamiliar questions that you might encounter in your upcoming exams. So this is for those of you who are preparing for the May June exams in 2021, and for those of you who will be preparing for exams beyond that, I hope that you find it easy to see how to approach difficult questions and how that might help you in answering them appropriately. So as you already know, paper 5, or as you might know, paper 5 usually has a lot of detail at the beginning, and it's very important for you to underline what the key things are. So here it says a group of students was given the task of planning a method to compare the effect of two different restriction enzymes on DNA extracted from peas. First things first, what are restriction enzymes? Restriction enzymes, and I'm just going to underline here using my red marker. Restriction enzymes are enzymes that are used to cut DNA. And if you remember from chapter 19, that is if you have completed the syllabus at your school, you will know that we often use restriction enzymes to cut fragments of DNA so that we can separate them either in gel electrophoresis or to do things like other kinds of genetic extractions or processes. So here it says the students used the following procedure to extract and digest DNA from the peas. 3 grams of sodium chloride was dissolved in 90 cubic centimeters of distilled water in a beaker. 10 cubic meters of liquid detergent was added to the salt solution and stirred. 50 grams of peas were ground up using a glass rod and then added to the salty detergent solution. The beaker containing the mixture of peas. So what I would say here is that when you're reading a question like this, try to sort of underline what the key things are. For example, liquid detergent, why would that be something that's added? Why were the peas ground up? Those are some of the things that I would want you to just put in mind as you read through a question like this. Then it says the beaker containing the mixture of peas and salty detergent solution was left in a water bath at 60 degrees Celsius for exactly 15 minutes. So left in a water bath at a fixed temperature. That's something else. The mixture was then cooled in an ice water bath for 5 minutes, stirring frequently. The mixture was filtered into another beaker. So out here, this is just things I would do with underline filtered. 10 cubic centimeters of filtrate was transferred into a boiling tube and 2 to 3 drops of a protease solution were added. So what is a protease solution? Protease is usually an enzyme that is used to break down proteins. So you can tell that there's some protein digestion that's happening here. Ice cold ethanol was then poured down the side of the boiling tube to form a layer on top of the filtrate. This was left undisturbed for a few minutes to form a layer of precipitated DNA. The precipitated DNA was separated from the cold ethanol layer by twisting the DNA onto a glass hook. The DNA was mixed with 20 cubic centimeters EDTA buffer solution. 5 cubic centimeter samples of EDTA buffer solution containing the DNA extract were placed into three separate test tubes, 1, 2, and 3. 2 different restriction enzymes, EcoR1 and HIND3, were added to the samples as shown in table 1.1. I'm now going to show you table 1.1. So this is what happened in the first test tube. As you can see, the EcoR1 was used. In the second test tube, HIND3, and in the third test tube, a combination of both of them. These were left for 3 hours to complete digestion of the DNA into fragments of different sizes. So remember that restriction enzymes are used to cut DNA. Then it says, this is the first question, and this is where I want you to start paying real attention or real close attention to what I'm doing. Suggest a reason for each of the following steps in the procedure for extracting DNA. Number 1, using detergent. Now, many students got this question wrong. That's one, because they were unsure of what detergent would do in breaking down anything or in basically playing a role. And I said to them, well, and I thought about it, like if you think about the fact that we mentioned COVID, because this is 2021, you've been asked to use soap and water to wash your hands frequently. What impact does soap have on the virus? What it does is that it breaks the cell membrane. If you think about the plants as well, they have a phospholipid bilayer. So you know that COVID has a lipid membrane. So does the pea plant or the pea cells. They also have a phospholipid bilayer. And what the detergent would do is to disrupt that cell membrane, so disrupt the phospholipids and make the membrane more permeable. So that would have been the accurate answer there. And again, just letting you see that sometimes it's not about having the technical knowledge, but associating other things that you know outside of the subject to the subject itself. Leaving it at 60 degrees Celsius, what would that do? Now we know that when you put cells at 60 degrees Celsius, you are going to denature most of their enzymes, especially if you are dealing with a eukaryotic cell. And in this case, what this is supposed to be, the answer here would be to denature the enzymes within the pea plants. And the reason for that obviously is that when you're extracting DNA, you don't want any other enzymes to be active because that might affect your extraction of the DNA. So say for example, you're extracting DNA, but RNA polymerase is active, DNA polymerase is active. That might affect the kinds of fragments that you get when you do add the restriction enzymes. The third one, why would you filter the mixture? It's important to filter the mixture because you're trying to remove the debris from the actual extract. So you're separating the DNA because the time when you filter the mixture in the procedure that was read out to you earlier on, you see that you had already done a lot of digestion steps and now you're simply trying to get the DNA out of the debris. And the fourth question here was, why would you add protease? And I think when I was reading the question, I said, you add protease because it is an enzyme that's used to break down proteins. And so just by saying breaking down some of the proteins in the cell, that would be the reason why you add protease. Then this is the question students are usually terrified of, the method question, because students often don't know what to write. And I'll tell you what I've told my students in class, and I feel like they've done really well because when they now answer the method question, they're more direct in how they answer and they avoid writing essays. So let's just read the question first. It says the students used gel electrophoresis to separate the DNA fragments produced by the digestion with the restriction enzyme. Describe the main stages used in gel electrophoresis that the students could use to separate and locate the DNA fragments. So already you know that you've done the digestion and all of those things. So you're simply preparing gel electrophoresis. This is my big advice regarding this question. Don't try to write it as an essay. And also avoid providing unnecessary information. So I tell my students to try to write one point separately, to write each point separately. So for example, if you are preparing gel electrophoresis, or if you want to carry out the process of gel electrophoresis, the first thing you need to do is to make the gel. And you know that the gel that you use for DNA is agarose. So you're going to prepare agarose gel, right? So just by saying prepare agarose gel, that would be 1 mark already. Okay, so prepare agarose gel. And then the second thing, when you prepare the agarose gel, if you remember correctly, hopefully you've done demonstrations in your classroom. If you haven't, I suggest you watch some videos on how to make an agarose gel. Is that you need to make wells within the gel. Wells where you're going to load your DNA sample. So you're going to prepare wells by using a comb. So you can say use comb to make wells within the DNA, within the agarose gel. And then you would speak about adding a buffer over the gel. The reason why we add a buffer is to maintain the pH of the agarose gel so that nothing dissociates with response to pH, and rather dissociations are due to size or charge. With DNA, it's typically as a result of charge and of size as well. And so we have the agarose gel, we've made the wells, we've added the buffer, and the next thing you have to do, because for gel electrophoresis to work, is that you have to plug it. So you then say you have to connect electric source, or in this case you can say pass an electric current through the gel. And again, I'll just write this. I typically tell my students to just write this. very simply in very short sentences, obviously not the way I'm doing right now because these are not sentences, but they would write very simply like this. And what this does is that it helps any examiner who's marking their paper to see very quickly what key points they are trying to make. It avoids them writing essays and simply trying to fill up the space with things that are irrelevant, because in the midst of so many irrelevant things, you might find that the things that are truly relevant are not even up to the marks that are required. For example, with this question, there are 5 marks, which means you need to make 5 different points. So we've made the well, we've made acarose gel, we've added wells to it, we've added our buffer, so add buffer. And again, you write this out in sentences. You don't write it out the way I'm doing now. I'm just doing this to the video doesn't take forever. So you add the buffer, and then you add the electric current or electricity. You can also call it the voltage difference or the potential difference. So electric current, and the fifth thing would then be you have to stain your DNA. So remember, it says something that you can use to separate and locate the DNA. Now, always remember that with gel electrophoresis, if you simply load the DNA without staining it first with something that is radiation, that can be read with radiation, you would not be able to see the DNA. So you stain the DNA and then you load it onto the wells, and you then be able to observe the DNA using a UV light, for example. You can also make reference in this question to a hazard or safety precaution, and there's usually 1 mark or 2 marks for that. If you say wear gloves, or wear goggles to protect your eyes, avoid irritation, all of those kinds of things give you marks. So it can be 1 mark or 2 marks, and just by writing all of those. So for example, the 5th one here would be stain DNA. But if you're feeling a little bit unsure, obviously, because they usually just mark 5 of whatever you write. If it's for 5 marks, if it's for 8 marks, they mark 8. And then you can just say number 6, you can add something about safety, and that would then give you full marks. So you write short, precise, direct sentences. Try as much as possible not to write essays in this question, because sometimes writing essays might mean that you're writing a lot of things that don't necessarily have to do with the question. So a dependent variable that the student could have measured or could measure. So in this case, remember, you're dealing with DNA. It has been extracted and it has been cut using restriction enzymes, and you've loaded that DNA. So think about your paper 5 as a progression of an experiment. So think of it as something that has begun, and then the following questions are sort of taking you through to the end of the experiment. So something that you could measure here, if you think about it, when you have gel electrophoresis, what you will get are bands, right? You'll get bands of DNA after the restriction enzymes have acted and the gel electrophoresis has been run. You get bands of DNA. And so what you can measure here would be the number of fragments of DNA from each restriction enzyme. That is 1 mark. You only have to write 1 thing, okay? Because it's just 1 mark. So just 1 thing is fine. Then it says, state 2 variables that the students should standardize to ensure the results from the different samples shown in table 1.1 can be compared. So I think there's the different samples there where it shows us like where we have the restriction enzyme 1 in 1 test tube and restriction enzyme 2 in 1 test tube. Well, something that you can standardize to ensure that the results can be compared with ease is you can standardize, for example, the temperature. So you can standardize the temperature. That's something that's very important. You can also standardize the current used for electrophoresis. You can standardize the time of the gel electrophoresis itself. You can standardize the amount of DNA that you add to the samples. So usually when we do gel electrophoresis, you would use a pipette, and that pipette would be set to a particular volume. So the volume of DNA is something that can be standardized just to make sure that the results from all 3 test tubes can be easily compared. I hope this is helpful. If you're not really finding it helpful, you can suggest a different paper that you might be able to work through. But I hope that this is sort of stimulating you to see how easy this question is, even if at first glance it might look a little bit difficult. Okay, so let's go on. Now again, always remember that when you're doing paper 5, you are progressing through an experiment. Even though it's not your experiment, you're simply role playing and sort of analyzing what someone else would do or what you would do if you were in the shoes of the experimenter. So now you have started with planning what you would do to set up the gel electrophoresis. You have also answered questions about the variables that would be standardized. You have answered what would be measured, which would be the dependent variable. Now you come to this point where they're showing you the results. And they say these are the results that you have gotten from this gel electrophoresis experiment. And if you look at these results, you will see here that cut width equal R1 are these two, the first two. So I'm just going to use the red marker at the bottom. So cut width equal R1, just going to circle that. So these two are cut with equal R1. These are cut with HIND3 and these are cut with both restriction enzymes. So basically there are two sets of images for each restriction enzyme. Then they say the students concluded, I'm reading from the bottom here now. The students concluded that the DNA of peas had more restriction sites for equal R1 than for HIND3 and that some of the HIND3 sites were within fragments produced by equal R1. State one evidence in this figure to support each of these conclusions. The first conclusion, that there are more restriction sites for equal R1 than for HIND3. That's very evident in how you see, in when you see these. So just look at here, you can see these white bands. These white bands represent your DNA. So I've had students referring to things like the dark regions or saying there are fewer dark regions in equal R1. Please rather refer to the bands that you can see, because that's the purpose of staining the DNA. If you remember from our method question, we said we have to stain the DNA. So you stain the DNA so that you can see the fragments, and these are the fragments that are now represented by these white lines. So what is the evidence that there are more restriction sites for equal R1 than for HIND3? First things first is that a restriction site is where a restriction enzyme would cut the DNA. So by looking at equal R1, you can see that it has a lot of DNA fragments, while HIND3 has fewer fragments relative to equal R1. So this is, that's a terrible drawing over there. Just going to, I just want you to be able to see what, so over here, these are the sites for HIND3, and you can see that when you count the bands for HIND3, they are less than the bands for equal R1. So just by saying there are more bands produced with equal R1 than HIND3 would give you 1 mark there. Then it says, the second conclusion, there are more equal R1 sites within fragments produced by HIND3. And the conclusion here that you can be able, the evidence you can be able to draw is that there are some of the fragments for HIND3 are not present in the mixed cut. So some fragments for HIND3 and for equal R1 happen not to be present in here. Over here, there are a lot more bands in here actually. So there are shorter fragments in here, which means that what this is telling us basically is that when we cut with equal R1 and HIND3 together, we are able to get more restriction sites. And so there is a possibility that some of the equal R1 sites are hidden within fragments produced by HIND3. Think about it this way. If you cut with only HIND3, your restriction enzyme would not cut at the sites for equal R1. Okay, but when you cut with both HIND3 and equal R1, you would be able to cut at all the restriction sites for both enzymes and so produce more fragments, and that would then be your evidence for that question. And that, just by putting that in, you would get 2 marks. Then it says state or suggest rather one reason why the results in figure 1.1 may not support the student's conclusions. Now, usually when they tell you to make suggestions like this, I often tell students, try to think back to what you know, especially regarding these experiments. So first things first is that if you look at these bands, in some cases, they are so close to each other that it is hard to distinguish them. I mean, for example, look over here. How many bands do we know that are in here? Yeah, we can see this one clearly, but is that a band? Is there a band somewhere in here? Because it doesn't look entirely black. It looks somewhat grayish. And so over here, you can then say like it's difficult to distinguish the bands because they're so close together. You can also say you can't tell if the extra short fragments are due to HIND sites, so HIND3 sites. So the extra fragments we see here, what if there are more HIND3 sites that have just come up for some reason? And also you can't tell where the extra bands have come from in this case, and you can't assume that they're from equal R1. So just thinking about like what assumptions can be made, because again the question tells you to suggest. So in this case you can think of the assumptions that you can make when you look at this data as an experimenter. What is it that you would not trust regarding this data that might make your conclusions falter? Okay, now this was the part that got a little bit murky for quite a number of students, so I think I'll just go through it, and it's the last part of the paper. And I know you're probably thinking, well, this is just one question, but it's taken so long. This is the thing with paper 5, it requires you to think really deeply. So here it says gel electrophoresis is also used in the sequencing of DNA. The method used for sequencing DNA is described below. A DNA molecule is first heated to separate it into two strands. DNA polymerase is then used to synthesize DNA using normal nucleotides and modified nucleotides. The 4 different modified nucleotides are each labeled with a different colored fluorescent dye. And you can see here it says red CTP, green TTP, blue GTP, and yellow ATP. The addition of modified nucleotides to the DNA strands stop the addition of any more nucleotides. And then it says here, this shows the formation of a 3 nucleotide fragment. And then you can see there, this is the original strand of the denatured DNA. And this is the direction of the replication. So the replication is moving in the opposite from 3' to 5', or from 3' to 5'. Then it says fragments of different lengths are produced depending on the position of the modified nucleotides. The DNA fragments synthesized are separated by gel electrophoresis and a laser light to identify the different colors. So now all that information we just read leads to this question. And this question is the one that had students a little bit confused because they thought, wait, what? So it says figure 1.3 shows the results of sequencing part of a DNA molecule. And then it shows us all of these fragments, and it puts a key here. And in the key, we can see again that we have CTP for red, and we have TTP for green, we have GTP for blue, and we have ATP for yellow. And we can sort of see those bands over here. So you can see those are 2 yellow bands in case that wasn't clear, and the other bands can be distinctly seen. Now, the first thing you need to recognize as a student with this question is that the very first letter of this tag over here is the nucleotide base. So this C over here, I'm just going to erase because I realized that I've messed that up. This C here is the nucleotide base that you're looking for. The TP can be considered some sort of fluorescent tag. Okay, and that's the same thing with this. So this is T, and this is G, and this is A. So basically, the red is C, cytosine, green is thymine, blue is guanine, and yellow is adenine. So now let's have a look at how we would write, what we would do with this information. It says use the figure in 1.3 to work out the sequence of the DNA template starting from the 3' end. Now, if you remember on the previous slide, the 3' end started on the right if you're looking at the screen right now. So it started on your right. That is the 3' end. But what I tell students when they're answering this question is to first write out what the full sequence is, just looking at the bands as they are. So let's see, yellow is A. So we have AA, and this is green, so we have AAT. Okay, so I'm just going to write AAT. Okay, so those are the first 3 bands that we have here if we're reading from left to right. And then we have our next set of bands. We have C here, we have T here, and we have G here. So that means we have C, okay? We have C, T, G. Okay, then let's look at the next set. We have blue, blue, and green. So this is looking G, G, T. So we can go there and just say G, G, and T. Okay. Then over here we have, what is this? This is red. So this is C for yellow, A, and this is T. So we have a cat there. So I'm just going to write C, A, T. And then we have more T. So this is T, this is C, and then we have two G's. So it is T, C, then G, and G. All right. So if you look at this over here, we have the template as the direction of, in terms of the direction of electrophoresis. So not the template, we have the sequence in terms of that direction. Now, what we need to do is to take note of what the question is asking. And this is where students often make a mistake, because I had students who got this right, but they were supposed to take a couple of steps further in order to fully get the mark. And what they had to read was, it says here, work out the sequence of the DNA template. This here is the sequencing, so the results of sequencing part of a DNA molecule. So this is not the template, this is the newly synthesized strand, which means we need to then do the pairings for each of these. So it means this needs to become C, C, G. Okay, this needs to become A, T. Okay, and we need to just go on like that. All right, and then have G, and this would become, I'm just going to write it on top, GAC, and this would become C, C, and this would be A, and then this would be G, A, and that would be T and T. So I'm just going to write it on top like that, T and T. All right, so that should be what you do next once you've gotten this sequence, because it tells you sequencing part of a DNA molecule, work out the sequence of the DNA template. That's the first thing. But the question doesn't end there. It says start from the 3' end. Now, if you remember on the previous slide, we looked at the 3' end and it was on the right. So when you're looking at the screen, it's on your right. So that means that you need to read this in reverse. So this then means, starting from the 3' end, that your answer here would be CCG. Remember, you're writing for the template, so you're writing the one at the bottom here. Then you're writing AT, and then you're writing GTG, and you're just going on until the very end. Sorry, not GTG. You're writing GAC rather, not GTG. I'm already making an error because I forgot I should be reading from the top here. So you're writing GAC, and you just go on to the end. And that is how you work out a question like this. I understand that it is a little bit heavy, but please just sort of take note of how it was answered, in case you never know with CIE, you might get a question like this just popping up in your exam. And just because it looks unfamiliar doesn't mean that it is hard. And this is something I just wanted to point out by choosing this particular paper, for students to become more confident with the unfamiliar and to just use whatever knowledge has been given in paper 5 to answer the question. I'm not going to answer the second question because basically it has been explained in how I have done the analysis for the first one. But I just want to point out to you again that with paper 5, most of the information that you need is already contained inside the question. And so always try to use your pencil to underline what the keywords are in every single question, so that you're able to draw on them when you need to answer the question. I'm going to stop here now. I know this was a little bit difficult and maybe a little bit too detailed for you, but I hope that you found it helpful. If there are particular questions that you have, again, please post them in the comments and someone from the learning community will be able to relate with you. Thank you so much for watching. Until the next video, have a good time. Goodbye.